Hello friends, this is Baswaraj Benangbe coming out with my next lecture video that is on organization of protein structure, which is helpful for D.PHARM and B.PHARM students. Before going into the different levels of organization of protein structure, proteins are made up of alpha amino acids. Why alpha amino acids? Alpha carbon atom, alpha amino acids. The carboxylic group and NH group are added to the alpha carbon atom, alpha amino acids. So proteins are made up of amino acids, which are polymers. Polymers means the many amino acids that are joined together. N number of amino acids are joined together by peptide linkage. So it is a polymer of go on joining amino acids. It will form a chain. So that is a polymer by peptide linkage. When you condense two amino acids, it will form a peptide linkage. When coming to the structural organization of protein, it is a complex three-dimensional rigid structure. We cannot write the structure so easily, protein structure. So it's a very complex three-dimensional structure. It is divided into four levels: primary structure, secondary, tertiary, and quaternary structure. Primary structure in simple way to tell, it is a linear chain or a straight chain of amino acids. You go on joining amino acids by peptide linkage, you will form a straight chain or linear chain. That is primary structure of protein. So in a simple way. So a polypeptide chain of amino acids joined together by peptide bonding, in which there is sequencing of amino acids linked by sequencing means different amino acids are joined together. Sequence means they are positioned at that particular number or position. Sequencing, it will form a chain by peptide bonding. And every protein molecule will have certain sequencing. Means every protein will have a chain of amino acids, and one particular amino acid will be positioned at that particular sequence. Sequencing will be there. Aspartic acid, then glutamic acid, phenylalanine. So whatever the different amino acids are there, so this amino acid should be there at the second position, third position, fourth position, fifth position. So on the different amino acids, the characteristic means that specific sequencing will be there. So if we change that sequencing, there will be change in physical or functional property of that particular protein. So that's why every protein molecule has to have some sequence. Means this amino acid should be there in this position. So that's all. The sequencing will be there. And presence of particular amino acid at specific position or number is significant. And in every protein, one particular amino acid will be there at specific position. It may be at 100 position or at 50th position. You cannot see that same amino acid in the other protein at same position. That amino acid will be there at that specific position in only one particular protein molecule. So that will represent the protein. So presence of particular amino acid at specific position is very significant, or number is significant. So if there is change in that sequencing of amino acids, if you change the sequencing, as I told you, there is a characteristic sequence. If you change that sequence, there will be change in properties. It may be physical or chemical, as well as there will be change in functions of the proteins. Okay, one example I have quoted here: single amino acid change leads to sickle cell anemia. Glutamic acid, which is in the sixth position, amino acid is in the sixth position in hemoglobin beta chain. But if this is replaced by valine, if you remove this glutamic acid and place it valine, then it will lead to sickle cell anemia. See, that's how it leads to a disorder. So change in sequencing of amino acid will affect the properties of protein or functions of proteins. So primary structure, as I told you, it is a linear chain. So the two amino acids are there. So OH from this and H, carboxylic OH and H from amine is removed in the form of water to form a peptide bond. CONH is a peptide bond. So there is a peptide bonding between. So one more amino acid will come. Again, this OH and H of that amine will combine. One more CONH will form. So that's how there will be a formation of chain. Linear chain, straight chain of amino acids by peptide bonding. You can see there. These are the two amino acids. There is a peptide bond. Two amino acids, peptide bond. So that's how you can go on joining there. You can make a chain polymer of this amino acids. You can see there. This is the peptide bond. CONH. This is one amino acid. This is one amino acid. CONH. Again, this is one amino acid. This is another one. There is a peptide bond. CONH. CONH. So that's how peptide bonding. So primary structure refers to linear chain or straight chain. Secondary structure refers to coiling. It will form a helical structure. So it is called as alpha helical structure because alpha amino acids are there. Alpha helical structure may be right-handed or left-handed. Local folded structure within the polypeptide chain due to interaction between. So when you make a helix, it should get some strength. You take a rope and make a coil. Can you make it? No. When you make a coil, there should be some strength should be there. Some interaction should be there so that that coil is strengthened. It remains as a coil. So for that, in this this coiling, secondary structure is coiling, alpha helical structure. That helical structure is strengthened by hydrogen bond. There will be hydrogen bonding between the neighboring amino acids or the neighboring amino acids. There will be hydrogen bonding. Hydrogen bonding occurs between carbonyl oxygen and amide nitrogen, that is amide nitrogen which contains hydrogen. This hydrogen will link with this O of carbonyl to form a hydrogen bonding. That's all. There are three types of secondary structure: helical structure, alpha helix, reverse turn, and pleated structure. Helical structure, as I told you, it will form a coil, regular coil. It may be right-handed or left-handed. That coiling may be right-handed or clockwise or anti-clockwise, in simple way to say. So that coiling is strengthened by hydrogen bonding, which I told you how hydrogen bonding. So that may be right-handed helix or left-handed helix. And the right-handed helix is more stable when compared to the left-handed helix, which you can find in nails, hair, and all this protein structure. So they more stable than left-handed. And a second structure, in case of secondary structure, second type is reverse turn or beta bends. There will be change in direction in folding. So, see, there is a coiling, clockwise coiling is there. Suddenly, at one particular position, there will be reverse turn. The coiling will take a reverse turn. So that is called as reverse turn or beta band. And such turn, reverse turn or change in direction or beta band occurs in where these proline and glycine amino acids are present. Why they only at these amino acids the reverse turn can take place or beta bands can take place? Because these do not contain any bulky side chain. Side chain is not there. Bulky side chain is not there. If there is bulky side chain, then it cannot fold. It cannot change the direction easily. Due to there is no bulky side chain in these amino acids, they facilitate folding. So that's why that reverse turn or beta bend occurs at in the where amino acid proline and glycine are there. And these bends can be strengthened by hydrogen bonding as well as ionic bonding. Different interactions are there which will give the strength to the protein structure: hydrogen bonding, ionic bonding, so that it will form a compact globular shape of protein molecule. And then the third type of structure, secondary structure, pleated. It is a peptide chain stretched and laid side by side, forming an hydrogen. Like just you place benzene molecules side by side, benzene molecules side by side. I'll show you the structure. And there will be hydrogen bonding between carbonyl O and hydrogen of amide. So the pleated structure may be parallel or anti-parallel if it is running in same direction, parallel or anti-parallel if it is running in opposite direction. That's all. You can see there. This is the alpha helix coiling of maybe right-handed. You can see there. There is hydrogen bonding between amide hydrogen and carbonyl O. There is an hydrogen. Wherever possible, there is hydrogen bonding between adjacent amino acid. There will be. You can see there is hydrogen bonding. So that's how hydrogen bonding takes place in alpha helical structure, that is in secondary structure. This is pleated structure. You can see this is like a benzene structure. So there is a hydrogen bonding between the amide hydrogen and carbonyl O. This may be parallel or anti-parallel depending upon the formation of hydrogen bonding. Pleated structure. The third tertiary structure is refers to formation of tight, compact, three-dimensional structure of polypeptide chain. Means further coiling, the compact coiling, so that we are trying to bring all the amino acids which are far away are brought closer. If any chance of any possible interactions are there, further strengthening is done, given to that protein structure in tertiary structure by different interactions like hydrophobic interaction, ionic bonding will take place, disulfide bonding will take place. So they will give further strength to the protein molecule by forming a compact three-dimensional structure. Such conformation in which all the interactions will take place is biologically active, and that protein in this in that conformation is known as native protein. Okay, so in tertiary structure, I told you the protein structure is strengthened by hydrophobic interactions, hydrogen bonding, ionic bonding, as well as disulfide bonding. Hydrophobic interactions will occur between non-polar side chain of amino acids. These hydrophobic interactions, hydrophobes are non-polar molecules and usually having a long chain of carbons that do not interact with water molecule. Good example of that is mixing of fat and water is good example of hydrophobic interaction. So hydrophobic interactions occur between non-polar side chain of amino acids. And if any amino acids which are having oppositely charged polar side chains, then there will be ionic bonding will take place. If there are two amino acids which are containing sulfur containing amino acids come together, that is which will have SH group, sulfidyl group. So SH group of one amino acid and SH group of another amino acid will join to form a SS bonding, disulfide bonding. So whichever possible interaction, it may be disulfide, maybe ionic, hydrophobic, or hydrogen, will take place in the tertiary structure, so that the protein structure gets the strengthened by these interactions. And lastly, the interaction between amino acids side chain helps the folding of polypeptide chain to form a compact structure of protein. Last level of organization of protein is quaternary structure. Many proteins are made up of single polypeptide chain. Many proteins are made up of single polypeptide chain. But some proteins are made up of multiple polypeptide. This polypeptide, one polypeptide, another polypeptide, 2 to 3 polypeptide chain will link together, will form a unit, subunit. That is one subunit. Multiple polypeptide chain forming one unit, that is called a subunit in quaternary structure. And such multiple polypeptide chains, such multiple polypeptide chains, when they are linked together, whole assembly is called as oligomer. Multiple polypeptide chains which are linked together as subunits are linked with each other, that you are calling it as oligomer. And when you form this such oligomer, even in that case also the strength to that particular protein is by the same forces, that is by hydrogen bonding, hydrophobic interaction, ionic bonding, and disulfide. Wherever possible chances are there, that bonding will take place. The same forces will strengthen the quaternary structure also. Okay. Each constant of a peptide chain is known as subunit or monomer, and that monomer may be similar or different in primary, secondary, or tertiary structure. So you can see that this is the tertiary structure, in which you can see there is alpha helix, pleated structure. If there is any reverse turn, that also may take place. But in case of quaternary structure, as I told you, it is a multiple polypeptide chains are linked together. This is one polypeptide chain. This is another polypeptide chain which will be linked. So this whole assembly is called as oligomer. Multiple polypeptide chain units are linked together. So that is quaternary structure of protein. Thank you for watching my channel. Please subscribe to my channel for pharmacy solutions. Thank you. Thank you.